Hello everyone. We'll continue with the mechanism of photosynthesis. Today we'll study the C4 pathway, which is also called as Hatch-Slack pathway. Now, C3 means the photosynthetic pathway where the first stable product of carbon fixation, C fixation is carbon fixation, is a 4-carbon compound oxaloacetic acid OAA. This pathway of carbon fixation occurs in the plants like maize, sugarcane, pearl millet, and amaranth. The plants which employ this mechanism of carbon fixation show the presence of two types of photosynthetic cells, that is mesophyll cells and bundle sheath cells. Now these bundle sheath cells are also known as Cran's anatomy. So what is this Cran's anatomy? The large cells around the vascular bundles of C4 pathway plants are called bundle sheath cells, and the leaves which have such anatomy are said to have Cran's anatomy. Cran's means wreath and is a reflection of arrangement of cells. These bundle sheath cells may form several layers around the vascular bundles. They have characterized by having a large number of chloroplasts, thick walls impervious to gaseous exchange, and no intercellular spaces. So I hope you understand what is Cran's anatomy now. Now these chloroplasts are dimorphic, that is, those cells and the mesophyll cells are granule and those in the bundle sheath cells are adrenal. So phosphoenol pyruvate is the carbon dioxide acceptor and present in the mesophyll cells, and the reaction is catalyzed by the enzyme phosphoenol pyruvate carboxylase, that is PEPase. The first stable product is oxaloacetic acid, that is OAA, which is a 4-carbon compound. Now this OAA is converted into malic acid and transported to bundle sheath cell where it is decarboxylated into pyruvic acid. The CO2 liberated is used for Calvin cycle in the bundle sheath cells. Pyruvic acid is transported back to mesophyll cells where it is used for regeneration of PEP, utilizing 2 molecules of ATP for each pyruvate. It is an adaptive mechanism to avoid photorespiration. The decarboxylation of malic acid in the bundle sheath cells maintain a high concentration of carbon dioxide near rubisco so that it functions as carboxylase on its oxinase and not as oxinase. So next is crescentinium acid metabolism. Among dicotyledon plants, the family Crassulaceae, that is jade plant and bryophyllum, and among cotyledonous plants, the members of family Liliaceae, Orchidaceae, etc. exhibit cam. The cam are characterized by succulent nature. Stomata open at night and remain closed during the day. CO2 fixation occur at night. That is darkness. PEP is carbon dioxide acceptor. Large quantities of malic acid is synthesized at night and is stored in vacuoles. Decarboxylation of malic acid occur during day and CO2 is generated. Released CO2 enters Calvin cycle, that is ribulose biphosphate is carbon dioxide accepted, and carbohydrates, sucrose and starch are formed. Drop in stored starch, millet occurs during night and day respectively. One thing here to be noted is carbon dioxide compensation point. The atmospheric concentration of carbon dioxide at which photosynthesis just compensates for respiration. Under such a condition, net photosynthesis is zero. It is higher in C3 plants than in C4 plants. So, CAM reaction mechanism you can observe in this diagram. During night and day, the stomata is open at night here. The stomata is closed during day. Atmospheric carbon dioxide forms oxaloacetate. Here you can see how the energy is reduced. Malate pyruvate forms two carbon compound. The rubisco content of chloroplast is 16%. This enzyme is found in higher percentage in the chloroplast. In green plants, for evolution of each oxygen molecule, 1 quanta of light is required. So, what are the different factors affecting photosynthesis? External and internal factors. Blackman's law of limiting factors. When a physiological process is controlled by a number of factors, the rate of reaction depends on the slowest factor. This means that at a given time, only the factor which is least limiting amongst all the factors will determine the rate of reaction. Now, the external factors are light temperature, light quality and light intensity influence photosynthesis. Light of wavelength between 400 nm and 700 nm is effective for photosynthesis, and this light is known as photosynthetically active radiation, that is PAR. As the intensity of light increases, the rate of photosynthesis also increases, but at higher light intensities, the rate of photosynthesis decreases. It may be due to two reasons. Other factor needed for photosynthesis may be limited, or destruction of chlorophyll, that is photooxidation. Now, temperature, photochemical phase remain unaffected by temperature, but the biosynthetic phase that involves enzyme catalyzed reaction is sensitive to temperature. It may be due to following reasons. At high temperature, the enzymes may become denatured or inactive. At low temperature, the enzymes may get inactivated. The carbon dioxide concentration in C3 plants, the rate of photosynthesis increases with increase in carbon dioxide concentration up to 500 microliter. In C4 plants, the saturation reaches at the concentration about 360 microliter. Soil water availability. Water influences photosynthesis in two ways. If water available decreases and plants show water stress, the stomata will not open and hence there will be decreased supply of carbon dioxide. Reduced water potential in the leaf reduces leaf expansion and reduced in the photosynthetic surface area. So this is the summarized graphical representation to understand photosynthesis. The different experiments conducted by various scientists. Then photolysis of water conducted in different plants, cyanobacteria, lower plants, higher plants. Then the entire photosynthesis in higher plants, the factors responsible for photosynthetic activity, the photolysis of water, the energy release, the compounds different in process of photosynthesis that happens in stoma and grana, different chemical compound found in the various units of photosynthetic elements. Here comes the summary of C3 and C4 pathway, formation of energy and different stable and unstable compounds, types of photophosphorylation. So you need to know the difference between cyclic and non-cyclic photophosphorylation. As we have already discussed, but these are the key points which will help us differentiate between non-cyclic and cyclic photophosphorylation. Cyclic occurs in thylakoid region. Both the photosystem 1 and 2 are involved. Cyclic, only the photosystem 1 is involved. Electrons expelled by the reaction center P60 do not return to it. But in cyclic photophosphorylation, the electrons expelled by the reaction center return to it. In non-cyclic photosynthesis of water occur, oxygen is involved. In cyclic, no photolysis of water and no oxygen is involved. So ATPs and NADPH2 are synthesized in non-cyclic, whereas only ATP is synthesized in cyclic photophosphorylation. The ideal conditions for optimum sunlight, carbon dioxide, and aerobic conditions, whereas in cyclic, it operates under low intensity of light, carbon dioxide, and anorganic conditions. So these are dominant in green plants, whereas cyclic occurs in green plants and photosynthetic bacteria. Non-cyclic is inhibited by dichlorophenyl dimethylurea, that is DCMU, whereas DCMU has no effective cyclic photophosphorylation. I will end up the class here. Thank you.